Good afternoon, everybody. I have the pleasure of introducing Dr. Caspar Chater, with whom I worked in ED in a collaborative project. And recently he was visiting Mexico. Then I requested him to give a talk here. He has happily agreed. Dr. Chater currently works as a MSCA Global Fellow in the University of Sheffield and UNAM combinedly. His work is mainly on stomatal development and its response to drought stress. He graduated from University College of London in 2005 with BSc Honours in Biology. He completed his doctorate degree in 2011 from University of Sheffield, and he studied evolutionary development of stomatal ABA signaling during his PhD. After that, he did a postdoc from 2011 to 2015 in University of Sheffield, and he was still concentrating on stomatal development, and that's where we worked in a collaborative project. He has been awarded Newton Fund for International Fellowship, and he also got Marie Curie Action International Fellowship, Global Fellowship in 2017, and he's working in that fellowship from 2017 till today, and he works both in University of Sheffield and UNAM, Mexico. So with that brief introduction, I request Dr. Chater to...
Thank you very much. A kind introduction. Thank you actually for inviting me to speak here today. I'm assuming this doesn't work and it's just this one, right? So I can... Oh, okay, yeah. Sorry? Okay, good. So, as actually I said, my work in Mexico over the past 4 or 5 years has been focusing on stomatal development, in particular with legumes. And this is really an ongoing collaboration between myself in Sheffield and Alejandra Covarrubias in UNAM. So this is still a lot of work in progress, and what I'm showing you today is some of it will be quite preliminary, others less so. So as you probably all know, stomata control water uptake and water loss by transpiration. And this largely occurs through mass flow. So you see in this picture here is a thermal image, so infrared thermal image of a moss spore capsule, and this is the moss gametophyte. And you see this cooler area in the moss capsule is where the moss has its stomates, its stomata. And I started off working on stomata in moss, as I'll go on to say, but the processes are the same, whether you're talking about a tiny moss with a few stomata on its capsule or a huge corn, a maize plant with millions of stomata. And apart from water loss and evaporative cooling processes, stomata are critical in nutrient accumulation, obviously CO2 uptake for photosynthesis, and also pathogen responses as sometimes the first port of call for many leaf pathogens. Okay, so this is a moss. This is the leafy tissue of the moss, and this is the sporophyte. This is the spore capsule of the moss. And you see this little bit here will come off and all the spores will dissipate. And this here is where you have the stomata. So you have water coming up from the leafy gametophyte, and you see a cooling of the capsule there at that point. And as I said, this occurs in all plants, and you have this kind of hydraulic continuum from the rhizosphere, from the soil, to the atmosphere, driven by these pores. Now, the plant can regulate water use and carbon gain in a short-term way and in a long-term way through modulation of the stomatal pore. So the short term is in aperture control, opening and closing the stomate on a minute by hour basis, depending on the environmental conditions. And then the long term is changing in epidermal patterning, epidermal stomatal density or size or clustering. So today I'm going to be talking only about this long-term developmental aspect of stomata. Now, in Arabidopsis, which is kind of the most known model for stomatal processes, what you have is in the immature leaf, this entry division into the stomatal lineage, and then a final symmetrical division to produce the mature stomate. Now, governing this transition towards stomatal cell fate are transcription factors called speechless, mute, and pharma. And they drive stomatal development. And then inhibitory pathways along this stomatal cell fate lineage are governed by epidermal patterning factors, which inhibit the transition states. So epidermal patterning factors are small peptides that are secreted by the cells and can then direct or prevent stomatal identity in the cells around it. And as you see here, by manipulating epidermal patterning factors, what you have are a whole range of stomatal phenotypes. And this is largely work done by Judy Gray at the University of Sheffield, as well as other labs in the United States and in Japan. And we have, if you overexpress EPF2, for example, you have very low stomatal densities. Whereas if you knock out or delete EPF1 and EPF2 and overexpress an EPFL9, you have very high densities. So these are useful tools for trying to understand the relevance of stomatal development in plants. So as I said, these are secreted peptides, and you have this protein conformation where you are cleaved here, and it's only the mature peptide that's secreted from the cell. And this is a cysteine-rich peptide, and these fine-tune stomatal patterning because you have a system whereby EPF1 and EPF2 are negative regulators, and they can then go attach onto their receptor, which is ERECTA, to cause a MAP kinase signaling cascade to inhibit these transcription factors. However, you also have a receptor antagonist, EPFL9 or stomagen, which competitively binds with ERECTA receptors to prevent EPF1 and EPF2 from transducing their signal. Now, this is a really ancient system in land plants. And my PhD work and my first postdoctoral work went on to show that in mosses, we have stomata in the sporophyte, which are governed, which are developed, which are produced by these same processes as in all other land plants. So this is a spore capsule of a moss, Fiscomatrella, and this is the capsule here, and you would have the gametophyte going down here. And these are the wild type like stomata on the base of that capsule. Now, if you delete a BHLH transcription factor, which is similar to the Arabidopsis, speechless, mute, and pharma, you completely remove the ability to produce stomata. And similarly, with the EPFs, if you delete a single epidermal patterning factor in this moss, you have this long ring of stomata because the inhibitory processes that prevent stomatal development are not there. So instead of having this nice regular patterning of the stomates, you have this profusion of stomata in long chains around the capsule. So it happens in moss, and it happens in our crop plants. It's a very conserved system. And because of this high conservation of stomatal development pathways, we can then go on to opening up possibilities of being able to exploit these processes to try to improve our crop plants. And this is where my work's recently gone towards, in kind of trying to translate these processes into rice, such as the work Akshay was talking about with Julie Gray, and now into legume crops. So I don't need to tell you anything about this really, but this is just the background of the research. Because of this massive demand in food that they're already facing, it's critical that we try and find new ways to improve our crop responses to water loss. And Julie Gray's group has looked at these EPFs not only from an Arabidopsis point of view to improve water use efficiency by reducing stomatal densities, but also trying to improve nutrient uptake processes and reducing stomatal density in barley, in wheat, and in rice. So her work has really been focusing on monocrop, on cereal crops, and showing that they can modulate water use efficiency traits by reducing stomatal density without having a negative impact on yield. And that's in bread wheat, it was published this year. And this is some rice work that we did with IRI. And you can see that even under water deficit stress, the plants that have lower stomatal densities can keep hold of that water reserve in the substrate for longer. So actually they can stay cooler for longer because they're using less water. Now, going back to the work in Mexico, so these are data from CIAT, and these are the historical bean yields across Mexico. Now, if Mexican farmers continue to use the same cultivars that they're using now, CIAT has estimated there'll be massive losses in yield across Mexico. Now, there are some areas where there are improvements, but that's because of the altitudinal differences, and therefore some areas that are too cold to grow beans now will actually be able to grow beans in a few years' time. But if we improve these beans to be heat and drought adapted, then we could potentially prevent this transition to negative yields on a yearly basis that we're seeing now. And this year has been particularly bad. As I'm sure you know, the rainy season was very long in coming, and it's had massive yield impacts, especially in the north, where a lot of bean growers are. So legumes are of particular interest for me because what we can see in cereal crops may not hold for legumes because of this nitrogen specialism that they have with symbiosis with rhizobial bacteria. So unlike cereal crops, leaf nitrogen is usually non-limiting to photosynthesis. And so assimilation is not proportional to leaf nitrogen in legumes. Now, if we reduce stomatal conductance in a similar manner by reducing stomatal density, we could have much greater water use efficiency gains than we're seeing right now in normal bean breeding. So here you see some work by Adams et al. A couple of years ago, last year, and current legume water use efficiency is pretty low by the standards that are set for non-legume crops. So there's a lot of work to be done in legume breeding that people haven't focused on because there's a lot of focus on non-legume crops. And this is the water use efficiency gains that could be obtainable if we close that gap. So one of the issues, and there are many issues with legume work, is that there are lots of paralogs compared to Arabidopsis and especially compared to moss. So my work has also been trying to understand what these EPFs do in common bean and in soybean. There's lots of duplication events, especially in soybean, that we have to try and work around. Here's Arabidopsis EPF1, Arabidopsis EPF2, and here's this moss EPF at the base here. And then we have stomagen, this positive regulator of stomatal development. And you can see with one stomagen gene in Arabidopsis, you have two in soybean. Now, a large part of the work has been looking at these genes and seeing what they do by complementing in Arabidopsis, but also we're moving towards looking at overexpression in soybean. Now, if you use the peptide, the synthetic peptide, we can get fairly interesting results by synthetic stomagen application to the pod or to the leaf, and you get increases in stomatal density as you'd expect from Arabidopsis. And if you apply synthetic stomagen to the bean, you have increases in water use because you have way too many stomata. But that's not what we want to do. So this is a paper by Akshay and others, and this is where he has deleted this stomagen gene in the rice, and we get consequently reduced stomatal numbers in the rice and improved water use efficiency. Now, what we're trying to do also is look at that in soybean, and this will be a collaboration with Andrew Leakey in Illinois, Tom Clementi in Nebraska, and Johannes Stuttmann in Germany. But these transformation processes take a very long time, just as they do in wheat and in maize. This is years' worth of work to try and see what we can come up with. But once we get these overexpressors, which should be early next year, we hope to then test them in elevated CO2, in water deficit conditions, and then hopefully in the field in face experiments in Illinois to determine what happens in a more kind of agronomic situation. And this, we hope, will be maybe completed, or early stages completed by the end of next year. And this is Brianda, one of our master's students, who's been helping looking at the common bean and soybean genotypes that we're making in Arabidopsis and next in soybean. But what about below ground traits? So obviously I said there's this continuum where you have the rhizobial material, you have the root material, and interaction with water use. What happens if we also reduce stomatal densities? Now, stomatal function and rhizobial nodulation are probably quite closely linked. Rhizobial-derived nitrogen is transported in the xylem. So if you modify transpiration processes, are we going to negatively impact nitrogen transport from the rhizobia? And this is something that's a big worry for us. Could lower stomatal density, lower stomatal conductance, improve water use efficiency, but at the same time make a crop that's really rubbish for the farmer? And drought-induced nodule senescence is a big thing. When you have drought, the legume gets rid of those rhizobia really quickly, and the nodules just senesce. So again, what will happen if we improve drought responses or improve water use responses to that mechanism? There are lots of things to consider. But EPFs, are they implicated directly in below ground processes? And this is where it gets slightly confusing. So epidermal patterning factors are primarily known in vegetative organs. So EPFL, as I said, is a ligand for ERECTA family ERECTA family receptors, but all the organogenesis and patterning in stomata and hydrathodes, we can also see other EPFs, not EPF1 and EPF2, but other EPFs involved in flower, anther, pollen, vasculature, other above ground developmental processes. So almost all the research over the past 10, 20 years has been looking at EPFs and ERECTAs in these above ground organs. Now, the work of Julie Gray and others at Sheffield show that EPF1 and EPF2 in Arabidopsis are not expressed in the below ground tissues in roots. But if you delete EPF1 and EPF2, or overexpress EPF2, you do have a root phenotype. So by increasing stomatal density, you can alter root area. So it's pretty much proportional. So if you have low stomatal density, you can have a low root area like we see here. Or if you have a very high stomatal density, like in the double mutant, you have a larger root system. Now, what they argue in this paper is that this is probably a hydraulic effect. You know, there are higher water demands, you therefore invest more energy in the roots, and vice versa. And in our rice overexpressors, so we overexpressed an EPF1 in rice, we don't see big changes in root architecture, but we do see massive changes in erenchyma. And you see here that there's an inverse proportionality between stomatal density and erenchyma. So what is going on there? It's too early to say, but we don't think that it's a direct effect. Mainly because by knocking out root-specific transcription factors, you can see similar effects because of this change in root architecture, then making changes in hydraulic demand. And if we try to detect EPF1 and EPF2 and EPFL9 in rice roots and in Arabidopsis, you never find it. It's either there in trace or not there at all. Now, this has been the story for decades now. So the earliest ERECTA work by Keiko Tori in Japan showed there was no expression in roots, there was no protein in roots, and since then more and more papers don't find EPFs or ERECTA in roots. But it is there, and it is doing something. Because two papers, one from 2013 and another 2014, show that ERECTA is present, very low expression, but it's present in Arabidopsis. So it's a case of what it's doing there, and why is it so infrequently picked up in transcriptomics experiments and protein level. Now here you have EPFL4 and EPFL6, and you see EPFL6 is expressed in the vasculature of the Arabidopsis root. And here you have a stomagen, or EPFL9, tagged with Venus, and you see it as well is expressed in the vasculature. Now, there is evidence in this paper that stomagen and EPFL6 are involved in cortex cell proliferation. So even if, let's say, EPF1 and EPF2 are not involved, there's still a possibility that overexpression, so constitutive expression of these EPFs, could alter the processes that are native to the roots. Now in beans, so this is soybean transcripts of different EPFs, and this is common bean transcripts of different EPFs in nodules and roots and root hairs. You see very low to non-existent expression of most of the EPFs. These two here are paralogs of EPFL9, stomagen, the positive regulator. So most of the time, you just don't detect them because either they're not there or it's trace expression. So to try and understand these processes before we see them in the stable transformants when we get them, I teamed up with a researcher in Phaseolus in north of Mexico City to do hairy root, so kind of stable transformations, but just in the root. So these are chimeric soybean plants, and we did it in common bean as well. So you see there's a root system that's transgenic, whereas the shoot is not transgenic. And then you just use an empty vector overexpressing GFP to detect which roots are transformed. And you then fuse your EPF2 with this GFP, again to see which roots are transformed in your construct, in your actual experiment. So this EPF2 was from soybean. We overexpressed it in the roots of soybean, and then a little while later, we infected it with bradyrhizobia to induce nodulation to see what we've got. So here is, you might not be able to see very well, but this is an empty vector expressing that GFP in the root system and in the nodules, and this is EPF2 plant, again, expressing that EPF2, which is fused with GFP. So you can see which roots are transformed and which nodules are transformed. Now, in terms of nodule number per plant, we have a small but not significant reduction in nodule number if we express this EPF2 in the roots. Remember, this EPF2 is not normally in the roots. And in terms of nodule area, there's a significant there's a significant increase in nodule size. So particularly if you have a greater than 4.5 millimeter area, you have a significant increase in the number of in the size of nodules per plant. This is strange, and we don't understand it yet. But if you cut into these nodules, and this is an empty vector nodule here on your left, and here's an EPF2 overexpressing nodule on your right, you can see that this is inside one of these infected cells. Okay, so this is a TEM of this area in the nodule. And this is a nucleus of one of those infected cells. Now in the EPF2 overexpressor, again you see can you see that? I can't see that. Has it died on me? No, it's still working. Anyway, you see these infected cells are much larger. And when you look inside the cell, yeah, now it works. Sorry. The nuclei are also larger. So how can we explain this phenotype? You have potentially fewer but larger nodules. And these nodules have got larger infected cells with larger nuclei. What's going on here? Now, the larger nodule nuclei suggest changes in endoreduplication processes. And Arabidopsis erecta is involved, we know it's involved in endoreduplication and cell size in above ground tissue. So if endogenous hairy root overexpression of EPF2 causes this, what would happen if we do the opposite, if we try and delete or if we try and use RNAi on EPFL9? Would we see a phenocopy? And that's what we're doing now. But going back to this ERECTA issue, ERECTA mutations can result in cell enlargement in cortex, but this here is in Arabidopsis above ground tissue, right? So you see wild type, and here you see ERECTA, and you see larger cells. But the difference here is this is a deletion of ERECTA. This is not an overexpression of EPF2, which you would think would increase ERECTA activity. So this is, it's very difficult to reconcile both results at this stage. But what it's probably got to do with is that you have ERECTA family and you have lots of different EPFs interacting with this ERECTA family of receptors. So again, it could be a situational thing where in one situation an EPF does one thing and in another it does something else. Now, ERECTA obviously has a very big history in wheat, and Graham Farquhar has a lot of very powerful work in the realm of ERECTA. But also, ERECTA plays a role in common bean. So this is a work of Matthew Blair and others from 2016 looking at ERECTA SNPs associated with arid zones across the Americas. And you see there are several SNPs associated with these more arid zones of Mexico, whereas other SNPs associated with these wetter regions of the Andes. So again, ERECTA is involved in leaf endoreduplication. We don't know if it's involved in the root or in the rhizobia. But we do know that it results in reduced stomatal densities. And this is where a lot of the water use efficiency work of ERECTA comes in. But to make things more complicated, the stomatal density phenotype of ERECTA in terms of crop water use efficiency doesn't come from direct involvement of the stomatal development program. Because what you have is changes in stomatal development, but not changes in stomatal index, i.e., it's a cell size thing. So what we're trying to understand now, and some work from a couple of Chinese groups, whose names I can't remember, I'm sorry, show that ERECTA is involved with another gene, another transcription factor called HDG11, which may be responsible for our phenotype in the soybean nodules, even though what we're manipulating here is EPF and not ERECTA. So what is the interaction between the EPFs, the ERECTAs, and the non-stomatal development program and the stomatal development program? It's still a bit confusing, I'm sorry. And then we're using non-transgenic approaches to try and tackle this problem of common bean drought responses in Mexico. So common bean, or Phaseolus vulgaris, the genus center of origin is southern Mexico, so probably Oaxaca, Chiapas. And its center of origin there then went on to go to South America and then go further north. And it was then domesticated in independent domestication events, both in North America, probably around Oaxaca, and in South America in the Andes. So you have different trajectories of domestication. But the common bean from Mesoamerica center of domestication is generally more drought tolerant than the Andean races. However, you also have in the north of Mexico, another domestication event of a sister species or a related species called tepary bean or Phaseolus acutifolius. And this was domesticated in the more arid zones, probably in Sonora, Chihuahua, etc. Now, tepary is a very kind of live fast, die young crop that's a drought avoider, and it produces lots of healthy seed under extreme water deficit. Whereas common bean, as many farmers know, especially this year, is not very good at water deficit response. So a lot of the work going on in CIAT, as well as groups like INIFAP in Mexico, is trying to understand the processes in tepary bean and trying to integrate these particular drought-resilient loci into common bean varieties. So my work's been more kind of transcriptomic at this stage, looking at differences in common bean and tepary bean leaf responses and early pod development in terms of terminal drought, which is what farmers experience most in Mexico. And we've got interesting transcriptomic data sets that are comparing and contrasting these species responses. And this is between Sheffield and UNAM again. Now, we're also screening EMS mutants that were developed by Tim Porch in Puerto Rico. And with the help of the Newton Fund, we are trying to find mutants in stomatal density related genes and then understand what's going on in terms of drought, in terms of rhizobial responses as well. So will reducing stomatal index and particularly stomatal density improve water use efficiency in beans? And it's still kind of early days by comparison to what people are finding in cereal crops. And could fewer and larger nodules actually be beneficial to the plant? And there's some evidence to say that could be the case. But these are complex processes and interactions between below ground and above ground parts of the plant, and EPF signaling is still a big unknown in some ways. But we hope that the more we look into this, the more we could fine-tune either using different promoters or different backgrounds for arid and semi-arid farming of legumes. And as I'm sure you will all know, there's now a lot of work going on to try and put the rhizobial symbiosis into cereal crops. And that is another step, another stage entirely to think about. But either way, we hope we're starting to unravel this water use efficiency and nitrogen use efficiency interaction. And before I go on further, I'd like to thank Alejandra Covarrubias and her lab and others in IBT, Guadalupe Zavala, Osvaldo Valdez in Fesis Tecala, and various funders for their support over the last few years. And on that note, I'm very happy to answer any questions.
Hello?
Hi.
Hi. I'm Gemma Molero. I'm working in wheat physiology here in CIMID. Very nice talk.
Thank you.
Sorry that I missed a bit of the introduction, so maybe some of the questions will be related with that. So first of all, in wheat, we usually, not only in wheat, in many different plants, we usually consider that we have 1-to-1 stomata ratio in the upper or above what's the adaxial and abaxial part, right? But this is not always true. This is not always happening.
No.
So regarding this, when we consider, so the interesting part of this different stomatal density will be also in the case of wheat when we don't work with transgenics that much to see or to screen for the different stomata density, right? If this can have some advantage under drought conditions. But the problem is that even though you have the genetic basis, you see a different pattern depending on the side of the leaf.
Yep.
How does it work there? The EPF factors, or is it differently regulated in the parts of the leaf, or there are other factors that actually are also influencing the stomata density?
It's both. So there's different things going on. There's the species, right? So some species don't have any stomata or the top or at the bottom. Then there's the heritage of that species or that cultivar or that elite line. Some crops have very high densities on the top and vice versa. It's difficult to say without actually looking at the spread in your population. But what is certain is that there are differences. If you have a germplasm bank, you will find some with very high densities on the top and very low on the bottom and vice versa. In terms of what's causing that, it could be any number of things. It could be nothing to do with the stomatal development pathway, and instead it could be interaction with light responses and light signaling pathways that then converge on stomatal development and cell size. It's very complex. We don't know is the answer.
Because, for example, in fact, it's funny because like 2 months ago I was reading a lot, even I read your paper, your rice paper.
Oh yeah.
And because I was thinking about how this can affect, because I saw in bibliography that many years ago in wheat, there were people actually checking stomatal density, and after in the field they saw no effect on yield. In your paper, with your rice paper, you see that when you reduce the stomata density, you actually have high water use efficiency, intrinsic one, right? The one that you represent, not the water use, right?
Right.
But also you see no effect on yield. So that's good on one sense because, okay, it's not affecting yield, but the farmers will think, what about if, for example, in the case of wheat, there is a positive correlation between the stomatal conductance and yield. And usually stomatal conductance is related with the stomatal density somehow, right? Mm-hmm. So the farmer could think, okay, in my drought scenario, I don't see a penalty on yield, and it's good because I'm saving water. But in my favorable environment, I'm limited because the plant has, when you have lower stomatal density, the transpirative capacity of the plant is affected, right? Yeah. So somehow the growth can be limited if suddenly the farmer receives some kind of rain. So this is some of
I completely agree. I mean, breeders have bred for high yield, which has been correlated with high transpiration rates, right? Which reduces water use efficiency, blah, 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 blah. And therefore you're also breeding for poor drought tolerance in some instances. If you look at a population and see a big spread in stomatal densities, there are many other factors that are playing that may not mean there's any relationship with yield. So only by looking at differences in stomatal densities within the same genetic background can you really pull those questions apart, I think. And that's what we're starting to do with these experiments.
Because I was thinking, and also because I'm involved in another project, but instead of reducing the stomatal density, is to reducing the sensitivity of the stomata to blue light. So what does it mean?
Right.
That instead of, okay, we keep the same number of stomata, but because we know that at some point, the stomatas, they keep opening without any gain in CO2 fixation, right?
Yeah.
So what do you think that will be the best strategy? Because when you make the stomata opening less, even though you have higher density, we are also, we can save some water, but also we don't have these more complicated structural changes. I don't know.
I think in terms of cereal crops, there could be massive gains for water use efficiency with changes in guard cell signaling. So improving response times to light, to vapor pressure deficit, etc. So I think that's really a great way that people are going forward in terms of transgenics. And there's some really promising work from Tracy Lawson.
Yeah, I'm working with her actually
exactly,
in that project.
Yeah. So yeah, I think that's going to hopefully yield good benefits. I don't see them as mutually exclusive strategies, though. I think if you could alter stomatal density and also alter guard cell responses, then that could also be a really, really useful tool, combining the two.
Because the concern with both strategies, and sorry, I'm finishing with this one, the concern with both strategy is the increase in temperature of the plant.
Yep.
In your paper, maybe I remember wrong, but I remember that at the beginning, your transgenic plants, they were warmer, but because they saved water, at the end, the picture that you show, they were cooler.
Exactly.
But maybe the gain was not observed because before, the stomata were closer. So, okay, at the end, they have some benefit, but because they don't take any benefit before, because actually they are warmer, indicating that the stomatas are, well, they are fixing fixing less CO2, so this is why we don't see the effect. And because so far in wheat, maybe Spanish and I still didn't read it, but so far in wheat, we have not observed experiments in the field with full root capacity. We still don't know how it's going to be the impact on field conditions.
Yeah.
This is still not.
I think temperature responses is a real concern, and there needs to be experiments to show that. Evaporative cooling processes are not just about leaf temperature, they're also about pollen viability and all sorts of other things that we need to look at, definitely.
Thank you.
Thank you.
I have a very general question. Your suggestion about reducing somatic conductance or density to improve water use efficiency, that's on the assumption that there's surplus capacity in the plant to conduct gas exchange or lose water and not take up carbon dioxide. Is that balance? One would think that with so much breeding and everything, that ratio would have been optimized. And what you're suggesting is that no, there is surplus capacity to take up carbon dioxide and is needed for maximum bioproductivity. Is that assumption?
I think that's a fairly good assumption. Not only because of optimization in breeding, but we are at a state where CO2 levels are increasing. So plants have evolved in a relatively low CO2 environment, and now they're being subjected to much higher levels of CO2, and therefore we probably have more leeway to manipulate these parameters.
Can I do just one very quick question? You only you only check the expression in the leaf tissues, but we also know that there are the tissues, even the stems, or in the case of wheat, the spikes, that they also kind of have some stomata. Did you actually check the expression in these other tissues? So we know that in the leaves, yes, in the roots, no, but what about in the stems, for example?
In the stems, there is also EPF expression for stomatal development. There's been some really interesting work in Arabidopsis as well, in fluorescence, showing lots of EPF-ERECTA signaling with interesting phenotypes. There is undoubtedly EPF specific to stem like HALA, which is an EPFL6, I believe. That's the stem lineage important. Yeah, there's EPFs. I think I showed that. Where are we? Maybe I can't find it now. Here we are. So yeah, flower, anther, pollen, ovule. It's all there. Yeah, and it's not just stomatal development. It's also cambium differentiation and vasculature.
Okay, thank you.
What changes in the various EPF transcription factors do you see during drought and heat stress?
So under water deficit, what the biggest thing the plant's going to do is reduce cell division. So actually all of the, or the majority of the transcripts of genes involved in stomatal differentiation are down-regulated because you have fewer cells entering the stomatal fate. Yeah.
And then after recovery from the drought episode, what happens then?
I haven't done this experiment, but I'm assuming once you have recovery, you have new growth and you'll have upregulation of stomatal development. So it's difficult to say with transcriptomics what this means because it's really about the number of cells entering the lineage rather than whether it's directly related to increasing or reducing stomata.
I have a quick question on your. Are you done, Sarah? Or you're going to follow up? Your transgenic with lower stomatal density being cooler at the end. How could that possibly happen?
So
cooling is a function of evaporation.
Yep.
So when you reduce the stomatal density, you would expect it not to cool as much as a wild-type plant.
but under water deficit conditions, the wild type plant is losing water quicker. So at the end stage,
I see, okay.
yeah. But you're completely right. Most of the mutants that have been picked out from screens are because of they're either hot or cool mutants, depending on the phenotype. Hi.
Could you estimate what the water loss through closed stomata is? So if you would assume that the regulation is really good and they would close all completely, does then the reduction in the density have a big impact then on the loss of water when all stomata are closed?
So if I understand you correctly, with all stomata closed, what is the difference in water loss between the wild type and the transgenic? I guess that would be the equivalent of nighttime transpiration, for example.
Or to put that question differently, is there a molecular adjustment in the behavior of stomata when you reduce density, whether they close just as fast as the wild type or they close slowly as compared to wild type? I think that's what Johannes is
Right.
probably pointing to. Do you know?
Just the short-term signaling override differences in density.
I think there's always this interaction. So you have, in high density plants, you have smaller cells, right? And with smaller cells, you could argue that you have faster stomata, smaller, faster stomata. If you have low stomatal density, you often get a compensatory larger cell. So it could be that it's a slower stomate, but I haven't done those experiments.
And my question goes into this direction of whether really restricting the overall potential of taking up the water, whether this maximum value, reducing this maximum value is really the thing what is required, or whether it's more the regulations are similar actually to the question before. So if they would have a lot of stomata but close quickly and really closely, whether this would have a bigger impact to adapt them.
I think it could be, and what you see in for example the tepary bean from the more arid zones of Mexico is that you have a higher stomatal density than common bean in the leaf, and they are obviously smaller and probably faster, but it's difficult to say without doing those experiments.
I have a final quick question for me, final, not for the other. The result on when you reduce the number of nodules, the size going up, you showed that the size increased, number was not statistically significantly lower. However, did you measure the total volume per plant? If the volume remained the same?
This is something we're working on with people in Center for Genomic Sciences. So we're looking at actual ureide concentrations. It's still not fully understood. Yeah.
Yeah, this resulted from your point toward this compensation. So normally, it's an age-old thing in plant breeding that you do something and something else goes up, or vice versa.
Yeah. Yeah.
Any last question? If not, then let's give a
Thank you.